Larissa.
Thank you very much, Peter. So my name is Larissa, and today I'm going to talk a little bit about my project. I gave it a bigger, easier title to understand, which is RED at the tumor nerve interface. And here I want to explore how the sensory neuron signals activate RED and then drive PDAC invasion. So the pancreas is it's formed by two main functional parts. So you have the endocrine pancreas and the exocrine. The majority of tumors arising from the pancreas will come from this region, the exocrine pancreas. And out of those types of cancers, 90%, over 90% of them are going to be ductal adenocarcinoma, or as I'm going to call that from now on, PDAC. In Canada, the numbers of PDAC are really interesting to look at. If we look at the incidence of PDAC, it is the 9th most incident cancer among males and the 8th cancer among females. And I thought that those numbers were really interesting. So it was estimated for last year, 2024, the 7,100 Canadians would be diagnosed with pancreatic cancer, and 6,100 Canadians would die from this disease. And that really aggressive type of cancer is also reflected in those numbers here, because even though pancreatic cancer is not among the most incident cancers in Canada, it is among the most lethal cancers in Canada. And that's also reflected if we look at the 5-year survival rate, which is less than 10%. And that can happen due to late diagnosis, limited treatment options, and also rapid and aggressive spread. And here I'm going to highlight the presence of this mechanism called peripheral invasion, or PNI. PNI is one of the ways where the cancer cells, the PDAC cells, can spread to other organs. So here we have some of the other pathways that the cells can use to spread. In blue, we have the adjacent organ invasion. We have in red vascular invasion, lymph node spread, here represented in green, and then in the yellow, we have the perinary invasion. PNI, if we look at the numbers of PNIs in PDAC, it is impressive. Some papers notes that PNI is present in 70 to 100% of the PDAC cases. Some other papers say that 100% of the PDAC cases are going to present PNI. And that can happen to due to the specific tropism of those cancer cells to the neurotrophic factors, and also to the complex neuronal environment that is present in the pancreas. So what is PNI? PNI is defined by the presence of cancer cells along and around the nerves. And those cancer cells, they can invade all of the nerve shells, such as the epineurum, which is the most external layer of the nerves, and also the most internal ones, as the perineurium and the endoneurium. And when we look at the data or the evidence about PNI innervation, how does the development of those tumors in the pancreas change the innervation, right? If there is a change in the innervation. This really cool paper just came out, not just in January this year, and they did this mouse model with where they showed that after they showed a difference in nerve density between the normal pancreas and the tumor tissue in the pancreas. So here we can see that the normal pancreas, we can see those innervations showing here. However, when there is the development of this cancer in the pancreas, it's clear that there's an increase in the innervation of the pancreas. And translating that to the patient, that increase in the innervation and PNI itself, will be correlated with pain, recurrence, and also an overall worse survival. It's also really interesting that although PNI has a really impactful clinical characteristics that we can see, It is still inconsistently classified, underreported, and also poorly integrated into the clinical decision making. So looking at PNI and PDAC, my question, or the question of the field right now, is how do tumor cells interact with the nerves and migrate along them? So here I have a really simplistic model of what can be happen. So we see the increase of the innervation in the tumor, and those nerves are releasing different types of neurotrophic factors. So here I just represented them in different colors. One of those neurotrophic factors that are being released in the nerve by the nerve microenvironment are called GFLs. And those are the glial cell line derived neurotrophic factor family ligands. And then those neurotrophic factors will activate a receptor in the cancer cells. So we see that this receptor could be mediating the interaction between the GFLs and the effects on the cancer cells. And you might not be surprised, but here we're gonna point to red as being this co-receptor mediating the signals coming from the nerves and then translating those signals into the different behaviors of the cell. So red is a tyrosine kinase. Its activation will happen after the formation of this complex, where we are going to have two red monomers coming together with two co-receptors and one ligand. After red activation, we're going to see the phosphorylation of its intracellular residues, and that's going to lead to the activation of different pathways, which we can include. We can cite ERK and MAPK, PI3K, AKT, and PLC gamma PKC pathways. The normal function of RADs being really demonstrated in the embryonic tissues, mainly in the development of the enteric nervous systems and the kidney, where RAD is shown to mediate or support axon guidance, neurite outgrowth, differentiation, survival, and synapse formation. In other tissues, RAD has been also demonstrated to support and sustain a subset of enteric, sympathetic, and sensory neurons. So I wrote this piece of data that I generated 2 years ago when I was interested into looking on RAD's effect on the neurons, on the sensory neurons. So those are sensory neurons isolated from a mice, and I wanted to see what happens to those neurons when you add a RAD inhibitor, in this case, abracanib. So it was really interesting to see that in the negative control, we see those neurite. I hope we can see it, but we can see those neurites. Yeah, thanks, Julie. We see this outgrow of the neurites. So they form those long and really branched neurites. We also see that after treating those neurons with GFLs. So we see the red is being active here and forming all those beautiful extensions. However, when we do add sapercatinib to those neurons, there is a drastic inhibition of the extension of the neurites. So that also supports what we've seen in the literature, showing the red is playing a role in this adult tissue, supporting this population of neurons. However, as I already mentioned before, the cancer cells can express RAD. RAD can also work as an oncogene, and that can happen through mutations or fusions that will result in a gain of function, or also through an increase in activity or expression of this receptor, of the wild type receptor. For my project, we're going to focus on the increase of expression of the wild type RAD, which has been detected in different types of human tumors, including PDAC, where it is correlated or it seems to contribute to cancer progression. In PDAC, RAD is expressed in 40 to 65% of the cases, and it is correlated with PNI, poor prognosis, and also shorter overall survival. So for the past years, our lab our lab has been interested in looking at rat's role in PDAC. So we have some data showing that the wild type rat, comparing the roles of cells with wild type rat and cells with a rat knockout. When we look at the wild type rat, we see that this is an spheroid assay, those cells are able to invade out of the spheroid, so we can see like all this movement of cells here. However, when red is not active in those cells, this invasion is inhibited. We can also, our lab has also demonstrated that red promotes proteolytic structures formation. So that's shown by those little black dots in the collagen. So when the collagen is being degraded, it's going to form those darker spots. And that doesn't happen when red is not active in those cells. And more recently, this is a piece of data from Julie's. We also see that red seems to be involved with cell motility. So here we have PDAC cells with active red just cruising around. They're going everywhere. However, when this red is not active, when we have a knockout cell line, it seems like those cells try to move, but they're kind of stuck. They can go too far. So having all this data together, we kind of put red in a position where it can contribute to tumor invasion through different pathways and different mechanisms. And here it can happen through cell outgrowth, regional invasion, and perinatal invasion, which is my focus. So looking at this background, here we have kind of two different fields. We have the cancer side of the project, looking at those cells expressing red and responding to GFLs. And we also have the innervation of the PDAC expressing and secreting all those neurotrophic factors. So my project and my hypothesis is that a rat would be in the middle of those two fields, like a bridge between those two sides, where rat can convert all of those nerve-derived signals, cues, into a cell behavior, which in this case, we've been exploring cell invasion. So just more like a formal hypothesis. My hypothesis that the neuron-derived GFLs activate RET on PDAC cells and then promote chemotatic invasion towards the nerves, and that would contribute to PNI. So this is my model. It might look a little bit overwhelming, but I'm going to break it in different pieces so we can go through it. I have three aims to test my hypothesis. Aim one, will focus on characterizing rat's contribution to PDAC invasion towards the neurons. So we're going to look at this phenotype. Aim two will focus on investigating rat's role in invadopodia maturation and ECM degradation. And aim three will look at the rat's role in actin remodeling and also focal adhesion dynamics. So let's start with aim one. So let's focus on this movement of the cancer cells towards the DRGs, towards the neurons. Before jumping into my aims, I'm going to talk a little bit about my experimental model. First, our PDAC in vitro model, I've been using two well-established PDAC cell lines, the PANC ones and the MIA-PACA2 cells. They both express RET in different levels, as we can see here, with the PANC, expressing more RET than the MIA-PACA cells. And we can see that at the protein and also at the gene levels. Thanks to Brandy, we have generated both PANCs and MIA-PACA cells with a rat knockout construct. And here it's just the validation. We have the wild type cells expressing total rat, and then two different clones of rat knockout cells with no rat expression, and the same for the MIA-PACA cells. And as Peter mentioned before, this came out of the paper that we published, just showing that alongside with the red knockout cells, I'm also going to use the pharmaceutical or chemical inhibition of red using selpercatinib. So that's the cancer side of the project. How about the sensory, the nerve side of the project? Our motto to explore more this crosstalk between cancer cells and nerves is I'm using the primary sensory neurons derived from DRGs. And DRGs are the dorsal root ganglia. You can be thinking, what is that? So the DRGs are those little structures that you're going to be find in each side of the spinal cord. And those, I like to see them as little pockets. They're full of cell bodies of the sensory neurons. So the cell bodies are all concentrated here, and all of the axons go into the innervating the organs, the peripheral organs, such as the pancreas. So what I did here, I isolated the spinal cord. I'm going to zoom in so it's easier to see. I isolated the spinal cord. I opened it in half, and then you kind of have to believe. You can see those little pockets here. Those are the DRGs. So I'll have one in each side of the spinal column. So from there, you have to cut them on the base of the nerves. So I will end it up with just the DRGs, those little pockets full of sensory neurons. So after that, we can use those DRGs for a variety of experiments. Here we established two different models. We did a whole mount DRG culture where I would make a matrigel droplet and then transfer this DRG to the center inside of the matrigel droplet. So I would create a matrix around the DRG. After leaving the DRG for 48, 72 hours, we can start seeing the growth of those extensions, and that's visible under phase contrast microscope, bright field microscope, but you can also use different dyes and fluorescent labeling to see them better. We also established a dissociated DRG culture, and here we use the same DRGs isolated previously, and we just did a series of enzymatic and mechanical digestion to obtain those two-dimensional culture with all the cells present in the DRG. So here I'm just highlighting the presence of the neurons. So the neurons are going to show the cell body is really rounded, so it's easy to separate them from the other cells. So after establishing the DRGs, we went ahead and start kind of playing with different conditions to co-culture those DRGs with cancer cells. So my aim one will pretty much focus on the use of the whole mount DRG plus PDAC cells. So after establishing those DRGs, I would just seed the cancer cells around it, and just follow it over time. So as Peter also mentioned, this protocol was just published, and it was a really fun experience. I got to do this work with one of my friends from Brazil, Erica, and some of you got to meet her. So it was a different experience. We had cameras involved. So now the protocol is published, and the video is also available, so you can watch the video of me demonstrating the main steps of the protocol. Okay, so now we have our model. We have the cancer cells. We have the DRG. My next step was to put them together and see what would happen. So this is one of the first images that I got out of this cold culture. So here in the middle, we have the DRG. Here we have the matrigel edge. It's really clear where the matrigel was sitting. I seeded all the cancer cells. They were GFP tagged, so they are in green here. And I used a beta-tubulin 3 antibody to mark the neurite extension. So we can see all this radial neurite outgrowth coming from the DRGs And we can also see the kind of the movement of those cancer cells invading through the matrigel towards the DRG. So this first step was really nice. It was really cool to see those photos showing that this model could be used to quantify this direct invasion of the cancer cells towards the neurites. And it's really cool if you look at higher magnifications and the same model. I might go even closer. Here we can see it's the same photo as I showed before. The DRG is sitting here. All the extensions are being extending out of the DRG, extended out of the DRG, and it's really cute to see the cancer cells in green kind of moving along those extensions going towards the DRG. And then we can go even closer and see chains in the cancer cell, one in co-culture with the DRG. So here we can see all those philipodias being formed and this more like this physical interaction between the cancer cells and then the DRG-derived cells. Okay, so far we saw that the cancer cells are invading towards the DRG. My next question here was, is that through chemotaxis? Is the DRG secreting factors? That's what I thought, but can I prove that the DRG is secreting factors that are attracting the cancer cells? So using the same model, I followed the cell movement over time, 0, 24, 48, and 72 hours. And it's really cool to see that there is a progressive presence of those cells in the matrigel area, demonstrating that the PDAC cells are invading towards the DRG derived signals by chemotaxis, that those DRGs are secreting those factors, and that's attracting the cancer cells towards them. And then is rat involved in that process? That was my follow-up question to that. And again, using the same model, here I had the Panc wild-type cells and the Panc's rat knockout cells, also over 72 hours. And we can see that after 12 hours, the cells start clustering around the neurites. They start coming together, and after 72 hours, the presence of those cord or chain-like structures are really visible. However, when we knock out rat in those cells, we lose the ability that those cells have to form those structures and to invade towards those cells, or at least this ability this is decreased. And there is a significant difference between the area of invasion when we compare the Panc's wild types and the rat knockout cells, also suggesting that rat activity is necessary for this direct invasion towards nerves. So far we've seen that the cells are moving towards the DRGs. The rat knockout cell seems to not move as much as the wild type cells. What happens if we do a mixed culture? If we have the DRGs and then we have a co-culture with the rat knockout cells and the rat wild type cells, how are they going to behave? So that's what I did here. Here I have it's not over 72 hours, so it's a little bit earlier. When I started seeing the formation of those structures, it's like the initial steps of invasion, I was interested to see who those cells were. Are they rat knockouts? Are they rat wild type? In my mind, they would have been the rat, the wild type cells. And if we look at the fluorescence image here, we have the Panc's wild type cells tagged with M scarlet, so they're going to be red, and the Panc rat knockouts, they are expressing EGFP, so they're going to be the green ones. And it was really interesting to see that the leading cells of this process, they were expressing rat and they had rat active in them. So they were responding to, again, the cues that the DRGs were releasing. So this piece of data made us think that rat might be defining a later behavior on those cells towards the DRGs. And that fits really well with the beautiful work that Julie has done in Boston. So Julie here, if you don't know her, she's here. Julie Julie looked at the invasive potential of those cells. So we have the wild type cells and the red knockout cells. Now they have different colors. So the wild type cells are the green cells and the red knockout cells are the purple cells. She made an spheroid with those cells and followed them over time to see if they were invading the matrix around them in different ways. So I'm going to play this video. We can see that there's a movement of cells in the spheroid, and then they start like extending those cell extensions and moving out. It's really like visible that they are moving and invading out of the spheroid. However, when we knock out red, those cells lose the ability to move out of the spheroid. So they're not even trying to go out. And then Julie also did a mixed co-culture with both cells, where she had the pancreatic types and the PDAC, the red knockout cells in the same spheroid, also trying to see if when they are together, how do they behave. And it's really interesting to see that we see really similar patterns where the red wild type cells, they move out of the spheroid way earlier, and in a larger number of green cells are going out of the spheroid compared to the red knockout cells. So those when we look at those data together, it was really exciting because we were not even trying to sync our experiments. So when we looked at them, we see a similar pattern where it seems like the red active cells, the wild type cells, they're not only moving or invading more, but they're also taking the lead when we have those two cells co-cultured together. And then to finish my first aim, I wanted to look at the dynamic of those cells when they are invading. Because so far I only had photos of a specific time point, but I had the feeling that the red knockout cells, they were invading, but they were not well organized and they were not able to form the same structures I have seen with the wild type cells. And that's what we see here. So this is over 24 hours. We see that the pancreatic type cells, they do cluster together to form those we can call highways towards the DRG. So we can see them. We can't. Yeah. We can see them moving. However, when we knock out red, it seems like the cancer cells, they come together, but they don't have a leader. They don't know where to go. So they kind of stay over there. And it was also interesting to see that when I added supracanine to the wild type cells, that phenotype that we saw with only the wild type cells was also decreased here. And on the bottom videos, there I wanted to see, is this effect mediated by the DRGs? And yes, likely yes. So when we don't have a DRG in the matrigel droplet, the cancer cells are not going to form those same structures, and they're also not going to move towards the inside of the matrigel. So here we can conclude that the red activity is required for this directional migration of the PDAC cells towards the DRG, and the DRG is also providing a guidance cue for those cells. So this is my my aim one, and we do have some more questions that we want to ask about this. And one of the main questions that I want to pursue now on this aim is, do red wild type cells, so far we've been seeing that they are the leader cells. Can they create those paths? And then the red knockout cells are using those paths to kind of also follow them. So that's where I am right now, trying to think about next steps for the aim one. So far we've seen this phenotype. We see that the cancer cells are invading, they're moving towards the DRG. And then I was interested to look at the mechanism after that, like, so why or how are the cells moving? So my aims 2 and 3 are going to address two mechanisms that can explain this movement, this invasion of the cancer cells towards the DRG. So aim 2 will focus on ECM degradation. It will investigate if red can contribute or mediate invadopodia maturation, and ECM extracellular matrix degradation. My first question here was, do the DRG-derived factors activate red? For this experiment, I used conditioning media. So the conditioning media, all the DRGs, I pretty much cultured the DRGs for 5 days. I saved the media that those DRGs were growing into, and then I treated my cells with this conditioning media. So every time we have a bunch of experiments using DRG-CM, so that's the conditioning media from the DRGs. And here I could demonstrate that the treatment with DRG conditioning media for only 30 minutes seems to increase red phosphorylation. And when I added DRG conditioning media to pre-treated cells with selpercatinib, that activation didn't happen. And can those factors promote invasion? So I did a transwell invasion assay, which even most of you are familiar with. But in this assay, we have 2 wells, an insert and a well. So here we're gonna coat the insert with Matrigel to just mimic an extracellular matrix. So the cells have to degrade this Matrigel to invade to the bottom well. And I seeded the cancer cells in the insert, and on the bottom, I have 3 different cell lines, which would give me 3 different conditions. So we did a indirect co-culture with HEKs and HEK cells, which we know that they do not express GFL. So here, red would not be activated. I also had a condition with SMC cells, smooth muscle cells, and they do express GFLs. So here we would have a red being active. And then I also had another condition where I seeded the DRGs, the dissociated DRGs on the bottom of the well. And here I could show that the cancer cells invade significantly more towards the DRG. So the PDAC wild type cells invaded more towards the DRG when compared to the negative control. And when red was knocked out in those cells, the number of cells invading was way smaller than the Miyapaka's wild type cells. Also suggesting that the DRG is producing those factors and signals that it's activating red and then enhancing invasion. So for the cancer cells to invade, they need to degrade the matrix. So the following experiment that we did was a DQ collagen, where we coated a slide with collagen and seated the cells on top of that. When the cells start forming those proteolytic structures and degrading the collagen, that's going to be fluorescent, so we can see in those green puncta. And here I did this assay using the Panks wild type cells. So here I had my negative control, only the Pank cells plus GDNF, so red active cells, those cells plus DRD conditioning media. So here also expecting that red would be active. And then I had a condition where I added DRG conditioning media plus the red inhibitor selpercatinib. So here we can see that there was an increase in the number of degradation spots or puncta per cell after treatment with DRG, and this was only after 72 hours. So there was an increase in the number of those degradation spots after DRG conditioning media treatment, and that degradation extension was reduced when we treated those cells with selpercatinib when red was inhibited. Also suggesting that red activation is promoting the ECM degradation by those cells. For my aim 2, my next questions involves a lot of what is being colocalized into the mature invadopodia. So I want to test whether red activation will increase the colocalization of a few invadopodia markers such as cortectin and TKS5 and WASP, and if that's and whether that colocalization will also if those proteins will also be colocalized with MMP14 in the mature invadopodium. And here I just brought with we have some evidence from our lab showing that when red is active in those cells, there is an increase in the colocalization between TKS5 and red, and TKS5 is a marker of invadopodium. So it seems like when red is active, there's an increase in the invadopodia formation. I just want to look forward into that and see whether the DRG conditioning media is also promoting the formation of those structures. I'm also interested to look to answer those two other questions. Does PDAC plus DRG conditioning media increase activity of MMPs, especially MMP2 and 9? So we can actually see the degradation of the extracellular matrix. And lastly, do the DRG conditioning media activate again the signaling pathways downstream red? So we are kind of going backwards here. So we have the cancer cells moving, and then for them to move, they have to degrade the extracellular matrix. So that's what my aim 2 is addressing. And for them to be able to move and degrade the extracellular matrix, they have to be able to polarize. So that's where my aim 3 is going to focus at cell polarization and change in actin remodeling and follicle adhesions in those cells. I have a preliminary piece of data showing that it's a cell motility assay. So here we have a mean square displacement analysis, and it pretty much tells you how much the cell is moving. So we have in red, the most motile cells were the wild type cells plus DRG conditioning media. So again, all the factors that the DRGs are secreting are stimulating those cells and making them to move more. And if we look at the green line, which is the wild type cells plus DRG, so they're moving more, plus uppercanib, now I turn off red, that motility, that movement of the cells go down and goes down to the same level as my red knockout cells plus uppercanib. So here we have a spider graph just showing the tracks and the trajectory of those cells under some of those treatments. We can see that the Miyapaki cells, the wild types with no treatment, they are fairly motile. So we see all the tracks. There is an increase in the motility of those cells when I treat them with DRG conditioning medium. And that is reduced when I add sulpercanib and I hit red in those cells. Also, again, suggesting that red is also playing a role in promoting the motility of those cells. I have tons of questions for this aim that I do want to explore. And some of them, maybe the first ones I will address in the future, is whether red activation increase the localization, the recruitment of some important proteins such as ARP2/3 complex to the leading edge of those cells. I'm also really interested in looking whether the DRG conditioning media, if this treatment would increase the follicle adhesion turnover in those cells and then making them to move more. And also looking back at the pathways downstream red, whether the red activity will increase the phosphorylation of some of those components. So here I'll bring back my final model. And so far, I'm pretty convinced, I guess, that the DRG-derived GFLs are driving this red-dependent and also directional movement, invasion of the cancer cells towards the nerves. And right now, where I am standing with my project is I want to see if the red activation happening here can mediate some of those molecular mechanisms, such as promoting the localization of the MMP14, MT1, MMP to the invadopodia, and then increasing the focal degradation of the matrix, and also looking at if red can mediate cell polarization and also the formation of protrusions after treatment with the DRG conditioner medium. And hopefully that process leading from seeing the cancer cells, seeing the cells moving towards the DRG, testing those molecular pathways, maybe some more, hopefully that would give us some insight of how PDAC and the nerve crosstalk is happening. I would like to just finish thanking everyone. I'd like to thank my supervisor, Dr. Dolores Mulligan, all my committee members, Dr. Craig, Dr. Nickel, and also Dr. Greer. I also would like to thank all the members of Mulligan's Lab. They're all amazing. And all my collaborators, because they helped me a lot to learn how to isolate the DRGs. And a huge thank to everyone who I always message, do you have animals? Do you have any spare animals? And they're always open to share some of their animals or just give their spare animals to me so I can do my project. Thank you.
Any questions for Larissa? Go ahead.
Great job, Olivia. It's really good. I have a question about the beautiful IF image that you got in your A1. You're saying how like the neurons branch out towards the tumor cells. If you see like have the DRG with the matrigel and no tumor cells around it, do the neurons still branch out?
Yeah.
Themselves?
Yeah, they do. So my timeline is I first establish the DRGs, and what that means, I wait for them to extend as much as possible before adding the cancer cells. So yeah, I usually, I'm gonna keep them 3 days in culture. That's usually enough for them to grow all those extensions to almost reaching the matrigel edge. And then after those 3 days, I'll add the cancer cells.
Okay, so the DRGs will grow regardless.
Yeah. Mm-hmm. Yeah.
You may.
Okay. I was wondering, in these PDAC tumors, is it common that you see different different levels of red amongst the tumor cells? So I was wondering how the wild type and knockout co-cultures like come to be. Like, is that what you see?
That's a really good question. I don't think we have information or data showing that there is a difference in red expression in the real tumor. I would say yes, just thinking about like how heterogeneous tumors are. Yeah, I'd say that probably there is a difference in level expression in those tumors. And yeah, I think that's how far I can answer that question. Thank you.
Were some beautiful images. Great talk. Would you mind putting up the image that you have of the collagen degradation and stuff?
Yeah.
Please? So in the last panel with the drug, maybe you already know.
Yeah.
Okay, so you indicated there's fewer, but obviously the punctate looks larger. Is there some sequestering going on or something that's still allowing aspects of degradation, but only in specific areas or other bits of information that you can comment on?
Really great question. So yeah, I also measured the area of degradation, and I didn't bring it here because I'm still working on that interpretation. But there is, even though there's a decrease in the number of degradations when we have DRG plus abracanib, we can see there's an increase in the area of degradation. And our first thoughts was that because the cells are not moving, they are kind of stuck there. They are maybe being able to form the initial degradation spots, but because they're not moving, they're kind of just degrading.
They're sticking.
Yeah.
There.
Yeah.
That's what I was thinking, but I didn't know.
Yeah. So yeah. So I'm working on more replicates to be 100% sure that that's actually a biological effect and all. Yeah.
It's great talk, really interesting stuff. What do you think is the kind of kinetics of the inhibitor in terms of how long you're expecting it to keep red activity low?
So for those experiments, for the long experiments, 72 hours, I usually refresh all of the conditions every 24 hours, just to make sure that it's all still, yeah, working. Yeah, because yeah, it doesn't go during 72 hours.
Yeah, I spotted that as well with the size of the areas of degradation, and I guess one thing to consider is trying to present it as kind of the percentage area of the cells. I think it's kind of a gold standard way to
Okay.
quantify these things, but yeah, it's kind of, curious and unsettling at the same time. You can get kind of completely different effects of the drug treatment if it's kind of the number of spots
Yeah.
versus the area.
Mhm.
I think there's still some biology to understand there perhaps, but have you tried the Mia-Paca cells in this sense?
Not yet, but they are also, yeah, we're working in this experiment currently. But yeah, we're definitely gonna try the Mia-Pacas, but we have to start with the Pancs because they're easier to image and
And then there's yields. No, go ahead. Immediately waiting. Have you tried to look at all the potential GFLs that you can quantify in these DRG conditioned medium?
we haven't, yeah, that's a really good point. Because there's no consensus about what the DRGs are secreting. They kind of all put them under the GFL's umbrella. I had an interest, I don't know if you remember, my first project was trying kind of focus on one of them, but that was becoming too hard to actually pursue and ask the questions. So we kind of stepped back and like, let's look at GFLs in total. But I still do believe that there is maybe a preferential expression or secretion of one GFLs than the other.
Yeah, I just wonder if you kind of know what the major GFL is that you're maybe reading out. I guess the question would be like, can you replace all the effects of the DRG on this migration or activity of these cells by just supplementing the media?
Yeah.
So you kind of lose the whole directionality thing.
Yeah. That's something we could try, yeah.
Sorry, I had a follow-up, Larissa, that got stimulated by conversations in the past with Dr. Morgan and maybe with you at committee meetings, but since you're taking the DRGs from the mice anyway, have you ever thought about taking that hunk of pancreas and sticking it under matrigel or isolating some of the nerves from the pancreas?
From the pancreas.
Correct.
I think that would be really cool. Not sure how, I don't have the skills to do that yet. But I think, yeah, that.
Say the isolation technique. Jesus. I mean, it's a beautiful image and it proves the point, but to Dr. Craig's point, maybe there are subtleties.
Specific, yeah. Yeah.
One answer.
And I do believe that there is a difference. So here, when I isolate the DRGs, I do a pool of DRGs. I choose a region, which it's more like the DRGs innervating the pancreas, but there are still a difference between the DRGs. So that's why when doing the first experiments with the cold culture, I think I've done 20 times at least to make sure that what we were seeing was actually a real effect.
Could you go back a few slides? I want to see the migration. I guess you used it, leukocyte or something for right there. Yeah. So in this experiment, the DRG effect is red independent though, right?
Yeah, there's still an increase in the compared to the rat knockouts, you mean, right?
Yeah. mean, if you're getting a migration, you're getting a change from here to here,
Mhm.
knockout cells.
Yeah.
With your
Mhm.
DRG, so it seems to be independent, whatever is driving that migration
Mhm.
from the DRGs is not RET mediated. Is that your interpretation?
I do believe that there's lots of things being secreted by the DRGs. And I don't think the RET is the only receptor being activated in those cells by the DRG conditioning media. So it might be that's the reason why. Because there is an increase when we look at the wild type cells with no treatment
Yeah,
and the
that's what I
DRG.
was sort of-
Yeah.
My brain kind of got.
Yeah.
So
there's
it
still-
seemed contradictory, this observation
Mhm.
versus, well, this.
Mhm.
Clearly, you're getting a DRG effect here,
Yeah.
but it's
Yeah.
inevitable.
I feel like when we knock out RET, there's a change in the basal motility of the basal speed of the not speed, but the motility of the cell. So the motility goes down when we knock out RET. And then we have in light green and light blue, we have both RET knockouts and the RET wild type plus supercatenated cells plus supercatenated plus DRG. So there's this difference here that's not RET.
Yeah.
Yeah.
So how specific is cell capping?
Sup-
Yeah, how specific is?
It's really specific to RET. It's a selective.
When they do kinome scan type analysis, it doesn't touch anything else.
That's a great question.
Pardon?
No.
Yeah, that'd be, I assume that that's been done at some point.
Mhm, yeah.
Because there are other kinases.
They could be also,
You might see some
yeah.
effects there as well. Okay.
So this single cell assay is, I guess because you're using the condition, you don't necessarily have that functionality.
Yeah.
But do you see any differences with or without the condition, on the direction of the systems?
I don't think I've noticed that, at least with this model, with the Mia packet cells, they usually move a lot. So the only things I could observe there was that they do move more when we add the conditioning media and kind of moving everywhere. I don't feel, and I feel like because the conditioning media is everywhere, there's no where where they could move towards.
Don't see any questions on the Zoom? Anything else? If not, join me in thanking Larissa for a beautiful presentation.
Thank you.